Hello everyone, welcome to my SCENIC video tutorials. If you are here to watch my SCENIC video tutorials, I believe you already know the function of SCENIC package in single-cell RNA sequencing data analysis. So I will give a very short introduction about the SCENIC package and also how to install the package. After that, I will show you how to load the data from different sources. So SCENIC is the abbreviation of Single-Cell Regulatory Network Interference and Clustering. It is a package to construct gene regulatory networks and identify stable cell states from single-cell RNA sequencing dataset. If you are a R user, you can install the R version, and if you are a Python user, you can install the Python package. To run the SCENIC package, the gene regulatory network is inferred based on the transcription factor co-expression with the target genes and also the DNA motif analysis. After the gene regulatory network analysis, the network activity is analyzed in each cell from the dataset to identify the recurrent cellular states. Because I'm an R user, so I'm going to show you how to install and use the R SCENIC package. To run SCENIC R, you need to install 3 other R packages: AuCell, R6target, and also Gene3. You can install those packages through BioManager install. AuCell is the package to calculate the regularly activities, and R6target is the package to provide the database for DNA motif analysis. Gene3 is the package to construct the gene regulatory network. You can also use the GRN Boost package instead of using Gene3, but this package is only available for Linux and Mac system. You cannot install it at the moment on Windows. So I installed the package already, so I'm not going to run those code on my computer. SCENIC 3 also need other packages. It depends on your installation for those packages on your computer. If you installed those packages already, then you don't need to install them. If you didn't, you may need to install those packages. If you don't have those packages, when we perform the SCENIC analysis, R will suggest you to install those packages if they are required. Another possibility, you can just click the package here to check if your computer has those packages already or not. So after install those packages, you can start to install the SCENIC package. You can install the SCENIC package through GitHub. Because we are going to use the demonstration dataset for the video tutorial, the demonstration dataset is a LUM file, so we need to install the scope LUM R as well. So you can try to install those packages on your computer. So next I'm going to talk about the SCENIC database. At the moment, the SCENIC database are available for human, mouse, and Drosophila data analysis. You can download the databases for human, mouse, and Drosophila. Use the link I provided here. We can also go to the website and have a look at the databases. So you can see here, this is the SCENIC databases. So you can see the human, mouse, and Drosophila databases. They updated the databases on 2022 in August. But at the moment, the SCENIC online tutorial is using the old databases. So you need to download from the old databases. So you can see here it's also updated on 2022 August. You can download the human, mouse, and Drosophila databases. For example, we can connect the human databases. It has both HG19 and HG38 databases. So we are going to use the HG19 databases. Then you can see the reference sequence are 45. Then it is the MC9NR folder. We need the gene bases. You can see it has 5 different databases. 500 BP up means 500 base pair upstream of transcription start site. Then they have the 10 KB around the transcription start site, or 5 KB transcription start site, or 5 KB upstream. Then the last one is 500 BP upstream and 100 BP downstream. You can download all of them, but for our SCENIC video tutorial, we are only use the 500 BP upstream and the 10K centered database. If you click the link, you will download the database. Once again, I downloaded all the database already, so I don't need to download them again. You can see from my folder, I have all the 6 target database in my SCENIC folder. So we can use those databases for our SCENIC analysis. So the size for all the databases are very big. You can see all of them are bigger than 1 gigabyte. So you can download the database. So next we can start to load the data. Because I have lots of videos to show you how to analyze single-cell RNA sequencing data using SURAT. So the first one, I'm going to show you how to load your data from SURAT object to perform SCENIC analysis. So let's use one of our SURAT object IPF fibroblast dataset. We can analyze the IPF fibroblast SURAT object as a SURAT object into R. You can see we have a large SURAT object here. So let's load the SURAT package first. Then we can use the dimplot to have a look at the cell clusters. You can see we have 5 cell clusters here. I named as myofibroblast 1, 2, 3, 4, and also the transitional fibroblast. So to get the data for SCENIC analysis, we need to get the array matrix data. For SURAT object, we can use the getCdata function from the SURAT package to get the array matrix data. The data is in the cRNA slot count. Let's connect the data set. You can see in the data slot says RNA and the count. So here is the count for each genes. So let's run the code to get the matrix data. You can see at the moment using the getCdata function, we have the expression matrix data as a large DGC matrix data. If you connect the data, you can see it's a S4 object of large DGC matrix data set. SCENIC requires the data set as a data frame to have the cells in columns and genes in rows, so we can change the large DGC matrix data as a matrix array. So let's change it. You can see now it is a large matrix data. We can connect the data set. You can see now we have the matrix array data. The count are around here. We have the cell barcode as column names and gene names as row names. So we have the expression data. Next, we need to get the cell types for each cell. We named as myofibroblast 1, 2, 3, 4 and transitional fibroblast. So let's get the cell type information from the root object. We can use the ident function to see the cell types for each cell type. Let's run. You can see the cell type for each cell. You can see, for example, this one is myofibroblast 3, this one is myofibroblast 4. So now we can get the cell type information from the root object using the ident function, and also we change it into a data frame. Let's run. You can see we have the cell information for our data set. Let's connect it. You can see the row names are cell barcode. We have the cell type information for each cell. So I showed you how to get the expression data and also the cell type information from your analyzed root object. If you are going to perform SCENIC analysis from your root object, you can get your expression matrix data and cell type information from your analyzed root object. In case you have a different data set, I'm going to briefly show you how to get the expression values and also the cell type information from different data types. So once again, I want to emphasize the data format. We already talked about it when we get the data from the root object. So you need to the expression matrix data. The preferred expression values in the expression data are gene summarized count. The row names are gene symbols, not gene IDs, and the column names are cells. And also you need to get the cell information. If you don't have the cell information, you may have to analyze the data set and identify the cell types. So next, I'm going to show you how to get the data from NCBI website, the Geo database. We are going to use the GSE 60361 dataset as an example to show you how to get the data. So first we need to load the libraries Geo query. Then we can get the data use the GSE 60361 as the link. You can see R is going to download the data in my computer. So let's have a look at the data set we downloaded already in my computer. You can see we downloaded both 60361 dataset. Here is the expression dataset, and we also downloaded the Excel dataset. The array information is in this txt data file. At the moment, you see it is a compressed data file, so we need to uncompress this dataset. So let's uncompress the dataset. Let's have a look at the dataset again. You can see now we unzipped it. It is a txt file. So next we can get the expression data from the txt file. So let's load the library data table, then read the expression data in. So we can connect the data set and have a look at the data inside this file. So you can see we have the count around here, and the column names are the cell barcode. But here you can see the row names are not gene names. The gene names are in the first column as the cell ID. So we need to make some changes again to get the gene names. Then we can get the expression matrix as a matrix without the first column. That's the gene names. Let's look at the expression matrix data now. You can see we have the barcode in the column names. We have the count here, but we don't have the gene names as the row names. So we can add the gene names as the row names. Let's remove the geo data first, and also have a look at the dimension of the data set. So you can see we have 19,972 rows and 3,005 columns. You can see our data file for the gene names. It also have 19,972 genes. So we can add the genes as the row names for the data set. So you can see now we have the gene names as the row names. And also we make the gene names as unique. It shouldn't be, because the row names do not allow repeated names. So let's run the code. Now we have the expression matrix data. We can remove the text file we downloaded in my computer. So next we need to load the cell type information. Because this is a publicated data set, you can see the cell type information from the published paper. The cell type information for this data set also is available from AU cell package. Because we installed the AU cell package, so we can just get the cell type information from the AU cell package. So the data set in the AU cell package is named as the mouse brain cell labels. So let's get the cell labels. Then we can read the cell labels in as a data table and change it to a data frame. So let's have a look at the cell information. You can see at the moment the column name is level 1 class. So we can give a column name as the cell types. So I showed you how to get the expression matrix data and also the cell type from geo dataset. You can also directly read the expression matrix data from cell range output files. We can use the dataset that I downloaded for human IPF data analysis. So let's clear those dataset from here now. Then we can have a look at the dataset first. You can see when you have the output files from cell range analysis, it will produce barcodes file, features file are genes, and also the matrix count. So we have 3 files here. We can read this dataset in as expression matrix data using the read 10 times function from the thread package. So let's run. You can see we have large GGC matrix data here. I showed you when we get the matrix data from thread object, you can use as matrix to change the dataset as array matrix dataset. If you load the expression matrix data directly from cell range output files, you have to analyze the dataset, or you can get the cell type information from somewhere. And also the single cell experiment dataset is very popular. Sometimes you may find the data that you are interested in a single cell experiment format. So you can load the single cell experiment library, then use the counts function to get the expression matrix data from single cell experiment dataset, and the cell information normally are in the code data. Finally, I want to show you how to load the data from NUM files. Because we are going to use the demonstration dataset from the SCENIC package, it is the NUM file. Then I'm going to show you how to load the dataset as the online tutorial. Because we are going to use this dataset for our video too, so let's clear the data we have here and also the UMap plot. So to get the data from NUM files, we need to use the package scope NUM-R. Let's load the package. Then we can get the NUM files from the SCENIC package to establish a NUM path. Now we can open the NUM files. Then we can use the getDGE matrix function from the NUM files to get the expression matrix data. You can see from here already we have 862 genes and 200 cells. We can use the dim function to see the genes and the cell numbers. You can see it is 862 genes, 200 cells. We can click the data. You can see here, column names are the cell barcodes, row names are the gene names. We have the count here. So that's the expression data. We also need to get the cell information. We can use the get cell annotation function from the NUM files to get the cell information. Then change the cell information as a data frame. Then we can have a look at the total number for each cell type. You can see we have 29 cells for the extra epidermal cells, 43 cells for interneurons, 14 cells as microglia, 55 are oligodendrocyte, and 59 are pyramidal C1 neurons. So we can save the cell information for video tutorial 2. Now we can close the NUM files. Because later we need to cluster the cells based on the regular activity in each cell, so we need to set the kernel variables for each cell type. So I copied the code from the online tutorial. You can see we only have 5 cell types here in this dataset. They actually have 7 cell types here. I believe those cell types are the original dataset. Then they make a small size dataset for the demonstration. So let's follow the code to set the kernel variables. Because we have 7 kernel variables here, and we only have 5 cell types in the dataset. So let's set the kernel variables for each cell type in the dataset. We can have a look at the kernel variables. You can see now we have 5 cell types here, and we give each cell type a kernel name when we plot the cells. If you see the red kernel, they are interneurons. So let's save the kernel variables. We can use the kernel variables in our video tutorial too. So finally we can make a plot to show the kernel for each cell type. So let's run the plot. You can see we have 5 cell types here, and here are the kernel we assigned to each cell type. Okay, so I'm going to stop here for video tutorial 1. In summary, I give you a very short introduction about the SCENIC package, and I showed you the code how to install the packages, and also I showed you how to load the data from different data sources. At the end, we load the demonstration data from the SCENIC package, and also we get the cell type information, saved the cell type information, we assigned the color to each cell type, and saved the color as the color variables. Then in my video tutorial 2, we are going to use this dataset and perform SCENIC analysis. So you can install the packages and load the room files. Then we are ready for video tutorial 2. So please subscribe my channel if you haven't to do so. See you in my SCENIC video tutorial 2.